Hello, my name is Michelle King, and today we will be talking about DNA mutation and repairs. So let's touch on the importance of mutations. Mutations are usually bad for individuals but essential for species development. Mutations lead to becoming more fit, some organisms procreating more than others, and most importantly, it leads to evolution. Next, we will talk about the different categories of mutations. Somatic mutations are not passed on generationally, but when a mutant somatic cell undergoes mitosis, it does pass that mutation on to its daughter cell. Germline mutations are mutant gamete cells that give rise to an offspring containing that mutation in their somatic and germline. In this presentation, we will go over a couple different categories of mutations. We will talk about insertion and deletion mutations, repeat mutations, forward and reverse mutations, suppressor mutations, and base substitutions. So let's talk about base substitutions. In base substitutions, one base becomes another. These are classified as transition or transversions. Transitions are a purine base changed to another purine base pair, or a pyrimidine base pair to another pyrimidine base pair. Transversion is when a purine becomes a pyrimidine. Let's talk about insertion and deletion mutations. We can call these indel mutations for short. These are types of genetic mutations that involve the addition, insertion, or removal, deletion of one or more nucleotide in a DNA sequence. So the insertion in the sequence leads to an increase of total number of nucleotides in the region. Deletion mutations involve the removal of one or more nucleotide, causing a decrease in the total number of nucleotides. Let's talk about frame shift. So that's an indel mutation that can cause a shift in the reading frame of the genetic code. This can result in a change in the way the sequence is read during protein synthesis, leading to the production of a non-functional or altered protein. This can have very big negative side effects. We have missense and nonsense mutations. Missense mutations are a change in the mRNA codon where a different amino acid is inserted. Nonsense is when a base pair change in the DNA causes a change in the codon so that an early stop codon is inserted. Let's now talk about the consequences of indel mutations. So, small indel mutations can disrupt protein function and potentially lead to genetic disorders. Large indels can result in significant changes in the genome, impacting genes and regulatory regions in the DNA. Please excuse my dog barking in the background. Insertion and deletion mutations, along with other types of mutations, contribute to genetic diversity within populations and across species. This helps with evolution because evolution relies on these mutations to introduce new traits. Then these traits are now subject to natural selection. Next, let's talk about repeat mutations. So, a repeat mutation is abnormal repetition of specific sequences of nucleotides that are in the DNA molecule. The most common ones are CAG, CTG, CGG, and GAA. In individuals with expanding repeat mutations, the number of repeats increase over generations due to errors during DNA replication and repair. So this means because the length of the repeat sequence tends to increase with each generation, it leads to more severe symptoms in the affected individuals of this mutation. So let's talk about some of the genetic disorders that are affiliated with repeat mutations. Myotonic dystrophy usually leads to weak muscles and myotonia, which is prolonged muscle contractions. Fragile X syndrome usually results in intellectual disabilities and behavioral challenges. ALS is characterized by loss of motor control, impairment of the arms and legs, which leads to things like tripping and falling, dropping things, and persistent fatigue in the individual. Huntington's disease usually leads to progressive motor dysfunction, cognitive decline, and behavioral symptoms along those lines. So, with repeat mutations comes genetic anticipation. Genetic anticipation is the tendency for symptoms of the disorder that comes with these repeat mutations to become more severe and start at an earlier age in successive generations of these diseases. Usually, these things can be diagnosed with laboratory testing, such as DNA sequencing and PCR techniques. This can be used to identify the presence of the expanded repeat sequences and specific genes. These tests aid in the diagnosis of the disease caused by these mutations. Now we will talk about forward and reverse mutations. Forward mutations is a genetic change that alters the wild-type phenotype of an organism, whereas reverse mutations is a genetic change that restores the original wild-type phenotype after the forward mutation. Forward mutations can be caused by various genetic changes such as point mutations, insertions, or deletions. Reverse mutations can occur through various mechanisms like spontaneous DNA repair or second mutation at the same site that restores the original sequence. Suppressor mutations. So these mutations essentially act as genetic fixes that can partially or completely reverse the impact of an original mutation. They can occur at different locations in the genome and work by either restoring the function of a disrupted gene or compensating for the defect in some way. I find these suppressors very fascinating because they demonstrate how cells can adapt to mutations and maintain the functions that are essential to living. Understanding suppressor mutations is crucial in studying genetic diseases and the mechanisms that underlie genetic variation. So let's talk about spontaneous versus induced mutations. So spontaneous mutations occur naturally without external factors or deliberate intervention. Examples include base substitution mutations and small insertions or deletions. Induced mutations result from external factors or deliberate actions. This is often used in research to study gene function or induce desired genetic changes in organisms. Examples can include X-rays, UV radiation, and chemical mutagens, like ethylmethane sulfonate, which is EMS. So how does our body detect these mutations? First, we have our proofreading during DNA replication. That's when our DNA polymerase goes through and has its proofreading functions and checks for the errors in base pairing and corrects them there. We have our mismatch repair mechanism, where specialized proteins detect and repair mispaired and mismatched bases after DNA replication. We also have our excision repair mechanisms, where enzymes scan DNA for abnormal structures, such as damage to bases or bulky add-ons, and go in and excise the damaged sections and replace it with the correct DNA sequence. So let's talk about how our bodies repair DNA. So we have BER. This corrects small sections of DNA, such as like single bases or a tiny section of bases. It removes the damaged bases and replaces it with the new correct one. We have NER. This repairs like bigger, bulkier DNA segments. These are usually caused by UV radiation or chemicals. We have our DSBR. This fixes breaks in both strands of the DNA double helix. We have our P53 protein. It acts as, let's say it's a guardian of the genome. It monitors the DNA integrity and triggers repair or cell death if the DNA is too damaged. Lastly, we have our telomere maintenance. This ensures the protection and maintenance of telomeres, the repetitive DNA sequence at the end of the chromosomes, to prevent genome instability. These mechanisms altogether help maintain the integrity of our DNA, preventing accumulation of things like mutations that can lead to diseases like cancer or other genetic disorders. Transposable elements, also known as TEs in genetics. So, TEs are sequences of DNA that can move within a genome. It changes position and can potentially disrupt nearby genes. So, let's talk about some of these transposable elements, the transposons and the retrotransposons. So, transposons move directly through and cut and paste mechanism, where retrotransposons use a copy and paste mechanism. Transposons encode enzymes like transposase that facilitate their movement. They also tend to cause small-scale mutations when they insert or excise. Retroposons include long terminal repeats and can lead to a larger scale genomic changes, including gene duplications. Approximately 45% of the human genome consists of these TEs, which really highlights the significance of them in our genetic makeup. TEs can also drive genome evolution by creating genetic diversity and contributing to speciation events. We use TEs in many different aspects, like in biotech, they're used in genetic engineering and gene therapy to introduce or manipulate genes in various organisms. So, in conclusion, mutations help with diversity and adaptation, where mutations introduce genetic diversity, allowing populations to adapt to changing environments and evolving threats. Mutations help with evolution because they're new raw material, which drives species' ability to develop new traits and characteristics over a long period of time. It helps with variation in genetics because genetic mutations provide variation needed for natural selection to operate, which leads to survival of the fittest. It also helps with the immune system diversity because, for example, in humans, mutations contribute to the diversity of the immune system, enhancing our ability to combat a wide range of pathogens. And it helps with biotech because mutations are harnessed in biotech for genetic engineering, gene therapy, and the production of new valuable proteins.